Mike or Caroline, one of you hosts, can you start the recording?
It's recording.
It is, okay. I've got into slide sharing and I've missed it. Okay. Well, good evening, ladies and gentlemen. It's about 7 o'clock. My clock is chiming, and welcome to tonight's Lifelong Learning presentation. Lifelong Learning is a free lecture series brought to you once a week, brought to you by the Professional Associates, a volunteer group here at the college, and President Stridicus's office. For the fall, last fall, as well as this spring, because of COVID and the rules on campus and safety of everyone, all of our talks are in a virtual format, and that we are using the Zoom webinar. To get into the Zoom webinar, you go to fortlewis.edu/LLL. But of course, you've already all done that, so you know that. But remember that website. Some of our presentations are recorded, some not. Tonight's is being recorded. So starting tomorrow morning, it'll be up on the college's website. And you could watch a recording or you could tell a friend to watch a recording. How do they get to it? They go to fortlewis.edu/LLL. And there will be a thing, watch presentation. So, or maybe it's watch recording, I forget which. But anyway, we are recording this. I want to tell you that next week, and that'll be February 25th, Thursday, Mark Swanson and Deborah Lycan are talking about decarbonizing transportation. So this is really a talk about electrical vehicles. We noticed today in the paper, I guess, that Ignacio is putting in another electric station. So we also put out a brochure. We have mailed those out already for the spring, but you could get a PDF. And in a minute, I will give you my email. And if you email me, I would send you a PDF if you don't have a copy of the brochure. But also Dick Grossman, another member of our team, every week he puts out a email announcing the talk and a little description of it. If you have been getting that, that's great. You're on our email list. If you're not, you can get to that. How? By going to fortlewis.edu/LLL. And way over on the left side, there's a place that you put in your name and your email address. That's all. And all you will receive then is the weekly announcement. Here's our protocol. We'll try to start this as promptly at 7 as we can. You folks out there are attendees and you're not visible. Your little thumbnail, other members of the other attendees don't see that, nor do the panelists, the speaker. As an attendee, you're also muted, so you can't speak to the other attendees, but we do encourage your questions. So please, during the talk, if you have a question, and hopefully at the end you'll have questions, you go to the chat button at the bottom of the screen and you can enter your question there. At the end of the talk, the speaker will address those questions, and we'll try to go offline certainly by 8:30. If you have any problems, comments, concerns, you can email me, Gary Rotman, or gsorcer@hotmail.com. We have a small committee. I've already mentioned Dick Grossman, Deborah Lycan. The speaker next week is one of our committee. Judith Reynolds, who started this whole program 21 years ago, is a member, and Chuck Carson. So now, without further ado, I would like to introduce tonight's speaker. Tonight's speaker is Dr. Caroline Kuleza. She is a associate professor of biology here at the college. She comes from back east, from the state of Maryland. She did her undergraduate work at Penn State, but then she got her PhD in microbiology from the University of Virginia. She then went and did a postdoc at Princeton. From there, she got a teaching position at the CU School of Medicine in Denver. And she taught microbiology. I think I mentioned her PhD is in microbiology. But in 2015, so 5+ years ago, she joined Fort Lewis College, and she is now an associate professor. Her research interest is really in something called HCMV. I will get in trouble as I try to tell you what that is, but that's human. That's the easy part. Cyto, which is, I think means cell, megalo, which I think means large, and virus. So that she does her research in the study of viruses. And those viruses that she studied, they can be life-threatening. That kind of sounds familiar, is what Dr. Fauci has been saying for the last year. And they're especially life-threatening in immune-compromised patients. So that all sounds familiar. We all understand that now. And the courses she teaches at Fort Lewis are in microbiology, topics in virology, topics in infectious diseases. So all of you that you can see fits in quite nicely with talking about COVID. So tonight's talk is called the COVID-19 Vaccines, Essential Tools to End the Pandemic. And now here, if we can do this, if I can stop sharing, we will have Caroline Kuleza.
Okay. Well, thank you very much for inviting me to talk to you all tonight. As Gary kind of told you in my bio, I'm a virologist by training, and my interests are in herpes viruses. I taught virology in the fall, and I teach microbiology now in the spring. And I kind of joke with students that it's a good year to be a virologist. That I get to apply all of the years of study that I've put in on viruses and share that knowledge with my students, my friends and family. And so that's been really rewarding for me. So I'm going to get started here, and I want to begin by sort of acknowledging a little bit about the current pandemic, right? We look at this as a historic pandemic of the 21st century. We're here 21 years into the 21st century. And certainly there have been, in the last 20 years, very significant epidemics, but nothing like this that most of us have seen before. And it's come with tremendous health and economic and social consequences, right? The upheaval in everyone's lives is something that we've been talking about for the last year. And so I just actually want to begin by kind of acknowledging the frustration and the heartbreak and the confusion and the anger that we sometimes feel over the pandemic. And I also want to say, though, that underneath all of that, I think there are really great reasons to be optimistic. And as a scientist, over the last year, I've watched tremendous scientific discoveries and innovations come out of this pandemic. And so I think there are, like I said, there are real reasons to be optimistic about the coming months and how this pandemic might resolve itself. So a little over a year ago, at the end of 2019, just to put us a little timeline, it was December 31st, 2019, that China alerted the World Health Organization to several cases of unusual pneumonia. And it was only a week later that they had identified the virus, SARS-CoV-2, associated with those unusual pneumonia cases. And then in February 25th of last year, of 2020, the United States CDC warned us that disruptions in the US from COVID could be severe, if only we knew, right? And then just a short few weeks later, the World Health Organization declares the corona outbreak to be a worldwide pandemic. And we've been off to the races ever since. I've put up some data from the Johns Hopkins dashboard. Today, currently, this is the latest update, worldwide, we're at over 110 million cases of coronavirus infections worldwide. And we're just short of 2.5 million deaths in 192 countries. That's the entire planet. Here in the United States, we have the unfortunate lead of cases and deaths, right? So we are closing in on 28 million cases of coronavirus infections in our country, and we're closing in on 500,000 deaths. I expect we'll probably hit that number next week or shortly thereafter. And so those are very sobering numbers. And when I as a community member, as a citizen, as a scientist, I've watched these numbers change over the course of the year. I watch them locally, I watch them nationally, I watch them internationally. And it's made me, and so I've reflected quite a bit on what I view or how important I think a strong pandemic response is. Let's see. And so we would like, I think in the ideal sense, we would like our pandemic response to be robust and strong, solid like a diamond, like a gem, right? And a pandemic response has a number of facets that are really important to all come together to make it that strong, effective pandemic response. And so I want to start by just kind of highlighting a few things that I think are really important facets of a pandemic response. Certainly, economic support for the entire enterprise of responding to the pandemic, for our health system, for our citizenry, right? The social and economic support that we need to kind of get through these times, to the economic support for research and development during this time is really important. Also, that we have robust health systems, right? That our hospitals and our medical providers are prepared and able to attend to large numbers of potentially sick people and to see them through to their recovery. This is really important too. I think we hadn't anticipated, you might be familiar with the idea of some folks who have suffered through coronavirus disease then have long-term consequences, or sequelae they're called. They're sometimes referred to as COVID long haulers. And that's something we hadn't anticipated that our health system might have to address in this pandemic. Also, really a central facet is a robust public health enterprise that's able to communicate risk to the public, to be able to manage testing, diagnosis, contact tracing, and follow-up. A well-trained public health enterprise is really important as a facet of our pandemic response. And then also, I think both our academic and industrial science activities worldwide play an important role in pandemic responses. For example, in developing pharmaceuticals, such as repurposing drugs that have been developed for certain kinds of infections, perhaps repurposing them for the current pandemic, developing antibody treatments, developing convalescent plasma. These are all an important role of that enterprise. And then lastly, and of course, why I've been kind of invited to talk to you today is a plan and the ability to develop and roll out effective vaccines against the infectious agent causing our pandemic, in this case, SARS-CoV-2. And so what I'm going to do today is kind of talk to you about the strategies that go into developing vaccines, and specifically about the messenger RNA vaccines. I'll certainly be happy to entertain questions about other vaccine platforms, but I'm going to focus on that topic. So in the process of meeting our pandemic challenges, we have some very specific vaccine needs, and I've kind of summarized them here in these 3 bullet points. When we want to go from having no vaccine to having a vaccine that is an effective facet of our pandemic response, what we're looking for, first and foremost, I think, is that we develop a vaccine that provides long-lived protection from disease. That is, at a minimum, what vaccines should do for us. Now, as an added bonus, we may have vaccines that reduce or eliminate the transmission of disease between individuals. But at a minimum, we want individuals themselves to be protected from disease. We also need those vaccines to be safe. As we know, now in the 21st century, Vaccination has become a little bit of a football, and the safety of vaccines has really come into question. And so being able to assure the public that they've been adequately tested and truly are safe, I think is an important thing that we need to check off when we're developing vaccines under in these kinds of conditions. And then finally, we're in this case developing a vaccine for a microorganism that has never been encountered by anyone else on the planet before December of 2019. And so we have a tremendous number of people who need to be vaccinated. So to meet that need, we need to develop vaccines that can be manufactured and delivered reliably and economically at a large scale. So, so what I'm going to do is I want to talk about, whoops, there we go. There's sort of two key elements that we need to understand when we're thinking about designing vaccines. And one of those big elements is sort of understanding how our immune system works and how natural immunity to pathogens occurs. And the other thing that we'll need to understand, I'll get to in a few minutes, is how the pathogen works. What does it do to cause disease in its host? And where does that intersect with our immune system? So briefly, and in one slide, I'm going to talk about how natural immunity to infections works. And there are entire courses, there are people who devote PhDs to learning about how natural immunity works. And so I want to acknowledge that it is a huge field, and I'm going to give you just a very small slice of how natural immunity works. So our immune systems are really amazing products of the evolution of the human species. And I kind of view the immune system as having two broad arms, right? And they are referred to as innate and adaptive immunity. Innate immunity is something that we all have that is really valuable on the fly. When you're exposed to some sort of pathogenic microorganism that can cause disease, we have an arm of our immune system that is there at the ready to try to deal with the problem. It isn't able to recognize what the problem is or adapt to the problem, but it's there as a first response. They're the first responders. So that's our innate immune system. But really what is the remarkable evolution in our immune system is this idea of adaptive immunity. And this is another arm of our immune system that really has two prongs. And those prongs of the immune system these two prongs do two different but interrelated things. And so in adaptive immunity, there's one prong that consists of cells of our immune system, blood cells, called B cells, and they produce antibodies. And I think a lot of us have heard about antibodies. They're in the news a lot, right, with respect to vaccines. So antibodies are proteins that are produced by our immune cells, specifically B cells, that these proteins help them recognize and attack invaders that are outside of cells. So when someone sneezes and you inhale some virus into your upper respiratory tract, that virus that's present could be stopped by antibodies that are produced by this arm of the immune system. The other arm of the immune system is a complex array of different kinds of cells, broadly called T cells. And T cells are able to recognize and attack infected cells. So once you become infected and you start to experience the symptoms of SARS-CoV-2, for example, you've got COVID-19, your respiratory cells are infected with this virus. And this arm of the immune response is able to recognize those cells, home in on them, and destroy them to limit the effect of the infection. So these together are our adaptive immune response, and is an incredible evolutionary feature of our immune responses. You've heard a lot, I'm sure a lot of you have heard a lot about antibodies and vaccines in the news. And with respect to respiratory infections, these antibodies and the activities of B cells are really an important feature of our immune response. I won't say they're the only thing, because this other arm, T cells, are really important too. But in the initial sort of development of this robust part of our immune response, the activities of B cells and their production of these antibodies is really important. So when we think about our immune responses and what vaccines are meant, what we want them to do, which is to ultimately educate our immune system to be able to fight off and prevent future encounters with a microbe. So in respiratory infections like SARS-CoV-2, right? COVID-19 disease is focused in our upper respiratory tract. If it gets very serious, it can descend into the lungs. And so that's what I've really indicated here with this orange coloring, that this is the site of infection, right? Of a naturally occurring infection. And then our immunity, our adaptive immune response, is targeted to that location in our body, right? That's where we develop immunity that can then fight off or prevent those future encounters. So the goal of vaccination really is to maximize the training of our immune system to elicit one of these, really the best response, right? Produce a lot of a good immune response in the location where we're at risk, and to do that in the absence of disease, right? To do it without the risks associated with infection. So I said to understand about designing vaccines, we needed to understand a little bit about the immune response, and I hope I've given you enough to sort of help us talk about this today. We also need to understand sort of the basics of infection with the particular pathogen that we want to develop a vaccine against. So, so let's look at SARS-CoV-2, which a little cartoon is over here on the left of the screen. And so the virus itself, the particles that are infectious and can enter your body and cause disease, are basically greasy little balls of protein. So there's protein and genetic material inside this greasy orange ball. And this greasy ball is studded with proteins on the surface. These look to me, I got this from NPR. I thought these were really clever cartoons about the virus, but these look to me like little Lego blocks, right? So these little Lego blocks stud the surface of the virus and they stick out into the environment, right? And so these proteins, and you may have, there's lots of reporting on this too. So you may have heard these referred to as the spike protein. It sounds very ominous. And the spike protein, when this virus enters our body in our respiratory tract, is capable of engaging at the molecular level with other proteins that are on the surface of our cells. And much has been made of this human protein on the surface of our cells called ACE2. So ACE2 is a protein, and it looks like a Lego brick as well in these diagrams, and it can engage in an interaction with the spike protein. So when that spike protein engages, you can actually also, a lock and key metaphor is an apt metaphor for this, right? This engagement allows the whole virus particle, this whole greasy ball, to merge with the target cell that it's going to infect and to get inside and then start the infectious process inside that human cell. However, right, because we are humans with highly evolved immune responses, our immune response is on the case right away, even as this virus is starting to initiate the process of infection. And so our antibodies, so our immune response can produce antibodies. And that's these antibody proteins that I've mentioned made by our immune response in this diagram are depicted by these little clear boxes. They look like little caps that fit on top of the spike protein. And when they do that, they get in the way of the spike protein interacting with its lock and key mechanism to get it into cells. So this is why I say, especially in respiratory infections, that antibodies are so important that we produce them because they help you clear an active infection, right? They are a key part of your response to an infection when you're sick. And by making those when you're sick, they help you prevent future infections, right? When exposures in the future. And they may even just help you reduce, if you're exposed in the future, reduce the severity of disease on second infection. So these antibodies are really important to our health. So how do we design SARS-CoV-2 vaccines? We know a little bit about how the immune response works. We know a little bit about the pathogen itself. And I want to reiterate that effective vaccines stimulate effective immune responses, which is why it's so critical to understand this aspect of infection. But they do that without the risk of COVID-19 disease, right? You don't have to run the gauntlet of severe consequences due to the infection. So in vaccine development, so I put on the screen here a very short timeline of the key things that have to happen in the development and rollout of vaccines in the human population. So this timeline, this gray bar, ends, it takes about, this says this takes about 10 months to a year and a half total. And I want to just pause for a moment to say that that kind of timeline is unprecedented. For COVID vaccines to be designed, tested, and deployed in 10 months, nothing like this has ever happened before. This is one of the amazing scientific innovations that has come out of the pandemic. To be able to deploy a vaccine on this timescale is simply unprecedented. We have never seen anything like this. Every other vaccine that you have ever taken yourself or taken your children have taken, this has been the number here is at least an order of magnitude different, right? Not 10 months, but 10 years, right? Minimum 10 to 15 years to get most vaccines designed, tested, and actively being used in the population. And so this accelerated discovery and testing process to me is just incredible. We are living in amazing times because this is setting us up to be able to do this for future pandemics and perhaps go back to other vaccines and redesign and develop them more effectively on a much shorter timescale. So this is really an amazing outcome of the pandemic. If you're looking for a silver lining, this is one of them. So let me highlight the steps here in the pipeline or in the timeline. So at the beginning of the timeline, you'll see I've got these mice here, and there's sort of two steps. They're interrelated, and there's the first steps, right? Right as we recognize that there's a new pathogen that we need a vaccine for, there are sort of the preclinical studies that have to be done, typically in animals, to establish sort of the form that your vaccine will take, what the platform will be to deliver that vaccine. Are your targets that you're using to stimulate the immune response actually effective at stimulating that response in animal models? So that's really the first step. You need a good candidate. You need a candidate that is likely to be able to move forward. Simultaneously, there's also what we call preclinical development or process development. And not only is it important to be able to design an effective vaccine, but you have to have the industrial and manufacturing processes in place, or at least starting to be put in place, to be able to produce that vaccine in large quantity and deliver it to the human population. And so now this involves a lot of immunologists, vaccinologists, if you will, virologists like myself doing basic science in this first step. And the second step is a really interesting group of people who have the scientific background, but also the industrial manufacturing background. I think they're the unsung heroes of our current pandemic vaccine rollout. So these steps are required to ultimately submit to regulatory agencies what's called an investigational new drug application. And if that's approved, right, if you have made a case for you've got something in your hands that could really likely be a good vaccine, then you can move into clinical trials. And I'll talk about those a little bit more later on in the talk. And then finally, the results of those clinical trials determine whether you can apply to regulatory agencies for emergency use authorizations. So I mentioned this idea at the beginning of the pipeline that you needed to identify the mechanism by which you were going to stimulate an effective immune response. And that involves what I would call a vaccine platform. And these, what I'm showing on the screen here, are the four most common vaccine platforms that are being used in coronavirus vaccines and also are being used for to develop other vaccines to other pathogens. You are probably yourselves most familiar with vaccines developed on the virus platform. And these consist of things called live attenuated vaccines. The measles measles vaccine is an example of that, or inactivated vaccine. Everybody who gets their flu shot every year gets an inactivated flu vaccine, although there are now some other platforms being developed for flu vaccination. These other platforms have kind of been in the background until this current pandemic. Nucleic acid platforms include DNA and RNA vaccines that I'm going to talk to you about how they work. But also coming online, companies are attempting to use what are called viral vectors as a platform. This is a platform where a different virus is used to deliver the information to educate your immune system. There's also protein-based vaccines, and we're currently using some protein-based vaccines, but this is a fairly new technology. It has a lot in common with, it sort of overlaps with some of these other platforms, but this is certainly a really interesting technology that is starting to be developed more thoroughly. But I was really asked to focus on nucleic acids, and we're going to look at the nucleic acid platform, the way to deliver your vaccine. Before I get into those details, I know probably a lot of you out there are aware of this, but I just want to draw your attention to the New York Times reporting on coronavirus vaccines. They have this page of their vaccine tracker, which I check out once or twice a week. I haven't updated this figure. This is from last weekend when I put this together. I added this up today, but I don't remember the number. It's somewhere around 90 or so different vaccines in the various phases of that pipeline, right? Some have already been abandoned, some have been approved, but there are a lot of folks out there, a lot of companies who are staking their reputations. They're excited about trying out new scientific approaches to really see if they can't add to the vaccine environment that we're in right now. So I highly recommend this site. The science reporting on these vaccines, how they work on the clinical trials is just outstanding, and it's really, really well written for a layperson. Okay, so let's talk about nucleic acids and sort of using that as a platform to deliver a vaccine, right? And so I know not everyone out there is a biologist, but I hope you got from your high school biology the understanding that there are nucleic acid molecules in our cells. And the two kinds of nucleic acids are DNA and RNA. And so DNA is that double helix, right? Those two strands wound together in a beautiful helix. DNA comprises our genetic material. When we talk about the blueprint of the human species, we're talking about the information encoded in our DNA genomes. But there is this other molecule called RNA, which less of us are familiar with. It happens to be one of my favorite molecules. I study it extensively in herpes virus infections. But RNA is a complicated molecule, right? It seems pretty simple. It's just this squiggly line here, but it has some similarities and some key differences from DNA. So you'll notice that it's just one strand. And although chemically it's similar, it's not identical to DNA. And what RNA molecules, they serve many purposes in our cells. But one thing that they do a lot of is to use that information that is in the RNA molecule to make the proteins of our cells. Proteins like antibodies, for example. Proteins like the ACE2 protein, the receptor for SARS coronavirus. So the information that is stored in our genetic material in our DNA is copied over or decoded into RNA molecules that then our cells can use to make proteins. It turns out an interesting discovery, laboratory science discovery, is that RNA molecules can be made in the cell, right, in our natural processes, and then they can do what they need to do, or in the laboratory, we can artificially synthesize RNA and deliver it into cells. And once we do that, it largely behaves like the naturally occurring RNA in our cells. Before I go too much farther, I want to introduce you to a scientist who I think is the unsung hero of RNA vaccines. And some of you may have read about her. Her name is Kate Carrico. She's been trained as a biochemist over the years. She's been very interested in the idea of how RNA molecules instruct cells to make proteins. And so for many years, she was interested in the idea of perhaps using the delivery of RNA molecules to cells to cure diseases like cancer, to any genetic disease, a chronic condition, by introducing RNA made in the laboratory into cells to reverse the negative conditions like chronic disease and cancer. And so for many years, she struggled with this problem. And she recognized that there's sort of two key problems, right? And the first of all, like this long squiggly molecule of RNA, messenger RNA, it turns out it's easily destroyed. It's easily damaged and can be broken into pieces. And when it's broken into pieces, it no longer functions in the way it should in cells. She also appreciated that if you just deliver RNA, messenger RNA into cells, on its own, it can trigger immune attacks on those cells. So you might want to try to solve a problem for that cell by introducing RNA, and you could make it worse because suddenly that cell would be vulnerable to attacks by your immune system. And so along with Drew Weissman, when she was at the University of Pennsylvania in Philadelphia, she made a major breakthrough in this field that kind of went under the radar for a number of years. And that breakthrough was using her training as a biochemist, she was able to discover ways to modify and make messenger RNA more stable, right? So that it didn't fall apart. It wasn't easily damaged by environmental conditions. And those same modifications made it less likely to trigger immune attacks, to trigger recognition by the immune system. So this is really an important breakthrough in the mRNA delivery field that was kind of under the radar until some other scientists who ended up founding Moderna and BioNTech, they recognized that, well, if we could deliver this to cells, we might be able to use this breakthrough in really interesting and important ways. So there's been some talk. I think Dr. Carrico is likely candidate for the Nobel Prize in Chemistry. That would make me very happy to see her hard work over decades of struggling to get her research funded and out there. It would be a really great reward for her at this point in time. So then how does this all come together in how messenger RNA vaccines work? So we have two parts. We've got our messenger RNA that's delivering some information to cells that's going to help them educate the immune system. And the delivery mechanism in the case of our current RNA vaccines are lipid nanoparticles. Both the Pfizer and Moderna vaccines coat these messenger RNA molecules that are delivered to the cell as the vaccine with these greasy balls. And I used that term before, greasy ball. That sounds a little bit like a virus, but this is really just a delivery mechanism. These little greasy balls fuse with the surface of cells in your body and deliver their payload, which is this messenger RNA. In the case of these vaccines, the specific messenger RNAs that are packaged in these lipid nanoparticles are then used by the cellular machinery to make a protein of the SARS coronavirus 2 spike protein. Remember those little red Lego blocks that we looked at on the vaccine particle? Those are actually depicted by these little, I don't know, they look like flowers or bombs. I'm not sure which. But these modified RNA molecules are delivered to the cell and they instruct the cell to make spike protein. I think actually I have a little there. All right, so these little flower bombs are the same as these little spike proteins, but they are expressed by this cell, stimulating an immune response to the spike protein. Remember those little clear caps that fit on top of this protein. And so an immune response to the spike protein ensues, developing antibodies that are effective at inhibiting its activity of blocking virus infection and replication. And this all happens without active infection, which is great. We don't run the risk of COVID-19. So if that's just generally how these vaccines work, I know that a lot of us have questions about, well, how do we know that they're actually safe? It's fine for that actually to work, but is it safe and how do we know they work? And so I want to highlight an important aspect of the vaccine development pipeline, which is these clinical trials. This would kind of be step 3. And there are clinical trials at phase sort of in increasing uh, detail and extent, phase 1, phase 2, and phase 3. And I want to focus on the phase 3, what are called randomized controlled trials. And they're often they're placebo controlled. So there is a placebo or a treatment meant to mimic but not produce the same result as the vaccine. So that's placebo. And they're often observer blinded. So even the people who are administering the trial don't know. Not only are they blind, not only are the participants in the study blinded to what they're getting, but the observers who are monitoring and running the study often don't know what participants are getting. So that's a RCT, or a randomized control trial. And so this is actually, I think, really amazing that we've been able to complete phase 1, 2, and 3 trials in such a short period of time, because we have a lot of skill now in designing these trials to run efficiently and effectively get to the results that we need, to answer the questions that we need to know. And I would say when you do, especially in these phase 3 parts of clinical trials, it's really important for the people running the trial to be very clear about what they're looking for. What is it that you want to achieve with the delivery of this vaccine? And so many times these phase 3 trials have very specific, what we call endpoints, right? And so in most of the phase 3 trials going on now, the endpoint is a question regarding, do we prevent symptomatic COVID-19? And that's it, right? We have lots of questions about these vaccines, but the simplest question that we can ask to see if they actually work and that they're safe is, how well do these vaccines prevent symptomatic COVID-19? During these periods, definitely lots of data is collected about their general safety. You've probably heard about how people have really widely variable responses or reactions to getting their vaccination. That's called reactogenicity. And so all those symptoms are cataloged and counted among the participants in the study to really document what we can expect when we might be actually receiving this vaccine. These trials in phase 1, phase 2, and phase 3 involve different numbers of people, right? The phase 1 and phase 2 clinical trials are focused on safety, and they're generally small, tens to hundreds of people, because the preclinical studies have already established some of the things to be looking for. And in COVID vaccine development, we've been really able to accelerate that because we have a lot of information about other coronaviruses and attempts to develop vaccines to other coronaviruses. So that those preclinical studies were really valuable. People studying SARS-CoV-1 starting back in 2003, or other emerging coronaviruses like MERS, which is Middle East Respiratory Syndrome. But these are all coronaviruses related to SARS-CoV-2. And so a lot of that preclinical and phase 1 data in other kinds of vaccine trials has been able to inform how to design and really get the most out of our clinical trials. But it's the phase 3, right, which is the largest trial. We have to enroll thousands of participants in this study to get to this question of what is called efficacy. Does this vaccine work? And specifically, we have to have an endpoint related to how that vaccine works. Do we want to alleviate severe disease, for example, right? These are different questions that have to be clearly delineated as the study is developed. So how do we know they work, right? And so we get these press reports on amazing vaccine efficacy from our mRNA vaccines. And I want to take a minute to kind of walk through this, a little bit of data. I was told I didn't have to show any graphs or data, but I want to show you these numbers because these are remarkable, and anyone can appreciate how incredibly efficacious RNA vaccines are proving themselves to be. So let me walk you through this. So you may have heard in reports that Moderna is reporting a 94.1% efficacy at preventing COVID-19 illness. That's their study endpoint that they were looking at closely in every participant of the study. But I want to encourage you, like, so, well, that sounds great, 95%. What about the other 5%? Are there 5% of people who are still getting disease? No. So let me I want to walk you through what it means when we talk about vaccine efficacy. And so what I'm showing you here in this little table on the right are the results from the New England Journal of Medicine publication of their phase 3 clinical trials. And I'd like you to focus first, let's focus first on the first column, which is the study group, over 14,000 participants who received the Moderna vaccine. They received the 2-dose vaccine and then were monitored. And out of that population of over 14,000, there were 11 cases of symptomatic COVID and 0 cases of severe COVID. Let's look over here at the other group, the placebo group, who received just basically a saline jab in their arm instead of the vaccine. So this is a similarly sized group. We're both over 14,500 participants. And in this group, we had 185 cases of symptomatic COVID and 30 cases of severe COVID. So altogether, what I'd like you to look at is just add up these numbers. So that's 215 versus 11. This is what we call we see the reduction in the risk, right? This is our risk of unvaccinated experience of COVID-19, right? That's the risk in the unvaccinated population. With vaccination, that risk goes from 215 in this population to 11 individuals. So that's where this number, 95% efficacy, comes from. We have a 95% reduction in the risk. And so that's really something to get excited about. Your risk may be higher or lower than this group, depending on how you compare to the individuals who were recruited for the study. But by and large, this group was meant to sort of encompass a large swath of the adult American population. So this is a really significant reduction in your risk when you take this vaccine. And that's true for the Pfizer vaccine as well. Your risk plummets when you take this vaccine. All right, I think that what I wanted to do also, in addition to talking about vaccines, right, something that's come up in the news in the last several weeks is the idea of variants of SARS-CoV-2 and the implications. How is that going to affect the vaccines that we're rolling out? Will they work anymore? Will they stop working? Do we have to make new vaccines? So I want to address that a little bit and sort of explain, talk a little bit about the emergence of these variants. So again, SARS-CoV-2, here's our greasy little ball with its spike proteins emerging from the surface. And the genome, the genetic material that makes up this virus, is subject to acquiring changes. We call those mutations, right? And so a variant has a mutation in its genetic sequence. In fact, it probably has several. And some of those changes in its genetic sequence change the way the spike protein looks. So that's meant to be shown by this change from the single little Lego block to the double Lego block, right? So this now there's been a mutation in the region of the genome that specifies the spike protein. And it looks a little different. Still gets this virus into cells, but this is different from the virus that antibodies might have initially been produced to interact with, right? So again, here's those antibodies I said, these little caps. Now these little caps don't quite fit on the variant spike protein. Now that might make the spike protein find its way a little quicker and a little more strongly to its target cells and get in a little faster. Or it might not, right? These are actually, it's without empirically testing them in the laboratory, it's hard to predict what the effect of these variants really is on how the virus is going to behave and how antibodies made to one variant of the spike will interact with a different variant of the spike. Generally, we would think that these antibodies can't bind to the variants, but that's not always true. And there's a lot of studies coming out looking at the antibodies made in response to our vaccines, how they are, we need to measure in the laboratory through some very sensitive assays whether or not they can still have an inhibitory effect on the variants. And I think that's a real big open question that's going to affect the trajectory of vaccine development in the next couple of months. Companies like Moderna and Pfizer, and actually any company that uses a nucleic acid platform, is actually poised to respond very quickly to make changes in their vaccine to address these variants. Other vaccine platforms won't be able to be quite as nimble. So there's other reasons. So that's another reason to be really excited about these mRNA vaccines. They're highly efficacious, and I think it's very likely that in the production process, we can respond to variants that cause trouble in the pandemic. So it's not always that these antibodies aren't able to bind. Sometimes they can. They continue to bind to the variants. Sometimes they bind with less avidity. So we really need to be able to, we need to do studies to figure out what is the case there. All right, so I hope that I've given you a broad sense of how messenger RNA vaccines work, some of the challenges that go into the vaccine pipeline, and all of the really amazing scientific discoveries and innovations that have come out in the last 12 months, just in virology in general and in the development of vaccines. It is an like I said, it's an exciting time to be a virologist. While it's hard to watch the struggles that we've had in the pandemic, to see these kinds of scientific discoveries come to fruition and really, I think, will make a substantial difference in the trajectory of the pandemic is so exciting, and I think is a really great thing to be able to be optimistic about in the coming months. So I thank you very much for listening to me talk about this. I'm gonna turn it back over to my moderators. They're gonna tell me how this works.
All right, let's see. I guess number one, we can stop the sharing or not.
Okay.
But all
Sure.
you folks out there, it's time now to ask questions. And then we'll have Dr. Kuleza answer them. The first one has been up for quite a while, but this is the different vaccines have to be stored at different temperature. Well, if they're both RNA or mRNA ones, what is it that's so different in them that makes one of them have to be stored at extremely cold and the other one at moderately cold?
So when I talked about Kate Carrico's work, I mentioned how prone to degradation, how easily destroyed RNA molecules are. And so you can store the colder temperatures that you store RNA molecules at, the more stable they'll remain. And that's true even for the modified RNAs that are present in the vaccines. They're then packaged into these lipid nanoparticles, right? These greasy little balls. So you have this mixture of fat or lipids and RNA. And they need to be mixed together in the correct ratios, and the RNA has to be inside the particle and it has to maintain its structure. And so in the empirical, right, or the experimental testing of these vaccines, those companies, Moderna and Pfizer, have decided that that's the appropriate condition to maintain the integrity of those vaccines. In practice, right, and those studies are like sort of very controlled laboratory studies. In practice, right, so I think more studies need to be done in practice, whether or not those vaccines can tolerate more extreme ranges of temperature or storage at slightly warmer temperatures for short periods of time. I think that's really going to become clear from information that comes from the actual deployment of the vaccine. Those conditions of extreme cold, ultra low temperature freezer, those were sort of laboratory decided or based on laboratory evidence, and primarily because of the nature, the weak nature, I don't know what to call it, the easily degraded nature of RNA. And I think those kinds of recommendations may change as we see like more deployment of the vaccine and see some extreme other kinds of conditions that the vaccine is exposed to.
I'm going to ask a question that it kind of comes in two different ways, but one of them is you just get a little tiny bit of the vaccine put in your arm. And if you're a 80-pound weakling like I am, or if you're a 300-pound person, why do you get the same amount of vaccine?
So in the clinical trials and in some of the earlier phases, in the phase 1 and the phase 2, those clinical trials look at different dosing, right? And so, so dosing, that kind of dosing isn't necessarily calibrated to your size or weight, but to your more to the stage, your stage in life and the maturity of your immune system. So you'll notice, you might have noticed that these vaccines are being approved mostly for people older than 16 or 18, right? Not for children, because there's a real difference in the maturity and the activities of our immune system. So it's really not so much that you're getting a dose appropriate to your weight, but you're getting a dose appropriate to the function of your immune system.
So this is the other side of that. So you take this little mRNA and you finally get a few of them or something. How do you go from that to be making millions of doses? And they're just running. What is the company doing? Johnson or Johnson or whoever does it? How do they take the sample that they made, let me call it the beta product, and then make gazillion of them?
Right, there is a pretty elaborate manufacturing process, and it's really interesting. I was reading about this recently, and it's really spread out in a couple of geographic locations. So the RNA is produced in the laboratory in a manufacturing process. Actually, it is derived from DNA. It is copied from DNA into RNA. So the first step is actually to produce a lot of DNA molecules representing your vaccine. So that's done in one facility. This is for Pfizer. So that's done in one facility, I think in Massachusetts or something like that. So they make all this DNA that's going to be used to produce a lot more RNA. And they make it, they put it in a case, they put somebody on a plane, and they send them to another manufacturing location to make the RNA. So it's kind of like a heart transplant. They get on a plane to go with all this DNA, and then they go to another location where they can scale up the production of the RNA that's actually going to be put in the vaccine. And then that has to be mixed with the lipid nanoparticles, right? So that all has to be materials that are sourced from other companies who are specialists in producing those kinds of supplies. And then there is a manufacturing process that is proprietary that I can't, it is opaque to me, but it takes mixing the RNA together with the lipid nanoparticles in particular ratios and under certain conditions that then produces large quantities of the vaccine, which then needs to be packaged and quality control analyzed, all that kind of stuff. So there's a tremendous manufacturing process underneath of that that's largely opaque to us, but that's kind of the outline of it.
So I want to say the number is now 38 million or so in the United States have had the vaccine, or maybe both of them. Are we being studied? In other words, is somebody doing the numbers on that? How many of us are getting it? How effective it is?
So I don't know. I haven't actually followed up on those kinds of studies. They're certainly, I can't imagine they are not underway, right? It's going to be really important to monitor in the broader population, right? Even though that randomized controlled trial of 30,000 people when they first were looking at the vaccine, that's a lot of people. There's still a lot that we need to understand, right? A lot of people have questions about, well, it prevents COVID disease, but can I still transmit it, right? Those kinds of questions have to be answered now when the vaccine is delivered to the broader population. So there certainly must be those kinds of studies going on, but they're in their infancy. I don't know a lot about how many are ongoing.
So another question is like, how do we know how long the efficacy will last? Or how do we know how they're going to be effective against the variants? I mean, is that going to come out of some other studies?
Well, that's going to come out of the studies that you're proposing, right? That we now have vaccinated 41 million people in the United States, either with their first or second dose. And among those, there will be subsets who will be studied to answer those questions. We'll look 6 months later or a year later. In the case of the first trials, right, there was not a very long period of analysis to ask questions about is protection, how long lived is protection? And that's a real question that we can't get an answer to until we have people who are vaccinated for longer periods of time. So all of us who are getting vaccinated, they're going to want people like us as we get vaccinated to get into those studies to exactly answer those questions. And the same thing about the variants. Like, we need to look at people who are vaccinated against the primary vaccines and say 6 months or a year down the road, ask about their exposure to the variants. Did they get exposed? Did they have disease? Were they protected? We won't know those until longer term studies are done. And I'm sure those studies are beginning to ramp up.
And kind of related to my earlier question, so I'm doing the Moderna, I'll have the second next week. And supposing then in June, I come down with COVID and I go out to Mercy or something. I mean, is that going to be a number in a study that says, here's a Moderna one out of the however many million that was, and they had both shots and yet they came down with it? Or
Yeah, so I doubt it, like, on a person-to-person basis. I don't know. There are probably some medical centers who want to do that kind of study. I don't know if, like, it would be a question of whether Mercy was participating in that kind of study. And if you showed up at their emergency room with those features, right? I've been vaccinated, but 6 months later I got disease. You might be enrolled in their study if they were participating. And I'm not, I'm not familiar with the kind, like, the participation of Mercy's medical staff in those kinds of studies. That would certainly be happening at bigger medical centers around the country.
so here's one, except it just jumped up. I have friends and family who are leery of the speed of development of this vaccine, and that this is a new type of vaccine, mRNA. Some to the point that they don't want to get the vaccine. And what would you tell them?
I would tell them that I know that it seems really fast, but in fact, mRNA vaccines have been studied and in the works, right? There's been no mRNA vaccine deployed, but there's been vaccines studied and in the works for well over 2 decades. And many, so the coronavirus pandemic presents sort of a unique problem because we have a population who is all completely naive, right? Nobody has experienced COVID. So it's actually really easy to design clinical trials to enroll a lot of people because nobody has had this disease before. Nobody is immune already. But RNA vaccines have been in the process of being designed and developed for other diseases that we do have vaccines for, right? So they've been studied for a long time, but oftentimes the economics involved in actually rolling out those, really bringing those products to market, has made it so companies usually have shelved them. But in this case, we have a novel pathogen for which there is no other vaccine, and there's an easy way to study it because we have a huge naive population. So it's allowed us to really do the clinical trial work on a very tight timeline. Let me also say that if you'll remember my timeline, I said there's a lot of preclinical and pre-process kinds of work that needs to be done. And a lot of that was already done for coronavirus, even though we didn't know about SARS-CoV-2, because there are a lot of people studying other coronaviruses and making vaccines to other coronaviruses. So that investment of the last 2 decades has really helped make that process much shorter than it would otherwise be. So I definitely appreciate that. That short timeline seems very strange. Like, were they cutting corners? But I would definitely reassure people that these clinical trials I think are quite accurate. We've had the appropriate size of the studies. And so the conclusions are robust. The safety is real, and the conclusions about how they work, I think, is accurate. And so that's what I would share with people who felt like that.
So we hear a lot about CRISPR technology, and what is the relationship between that and mRNA? And or sometime soon we're going to have vaccines that are made with CRISPR technology.
So I don't think CRISPR technology has much bearing on vaccine development in the short term. CRISPR technology CRISPR is a biological pathway discovered in bacteria and then has been applied to a lot of biomedical research problems. So CRISPR allows you to make genetic modifications and sort of and very specifically kind of study those modifications that you make. So I don't know that CRISPR has direct applications in the immediate development of vaccines. It certainly has some applications in the study of viruses and how they cause disease and how the immune system works. But it's really it's a technology that's employed to make changes to genetic material. And that's not really connected to how vaccines are developed or how they would be used.
And this question, I'm not sure I understand it. It's missing a letter, but what might be the prognosis for an all coronavirus vaccine?
Yeah. So wouldn't it be great? We have coronaviruses circulating in the human population that cause colds, common colds. A lot of kids get these other coronaviruses that are circulating in our human population. And then, of course, on top of that, we've had this coronavirus pandemic. And so the vaccines that we have like for Moderna and Pfizer, for example, right, are targeted specifically at SARS-CoV-2. But it is theoretically possible to create a vaccine that would be what we would call pan-coronavirus, right? That could train your immune system to recognize not only SARS-CoV-2, but MERS and other common cold-causing coronaviruses. I think the mRNA, the vaccines built on the genetic platforms of DNA or RNA, nucleic acid platforms, are probably the most likely to be able to produce that kind of vaccine. But I think we're a ways away from doing that. The goal right now is to bring SARS-CoV-2 pandemic under control. And I think for the short term, for at least in the next the next 2 years, right, the vaccines are going to be focused solely on controlling SARS-CoV-2. But some of the vaccine platforms could be adapted to target more viruses in one vaccine, which would be really amazing.
And this one, I don't know, you touched on a little bit, but why are children under 16 unable to be vaccinated?
So they were not so children were not evaluated in a randomized controlled trial, right? So we don't have data on individuals under the age of 18, essentially, right? Those were not participants enrolled in the study. And so we cannot make what is called the extrapolation, right, of the outcome of the the outcome of the vaccination in adults over the age of 18, right, who have what we would call mature immune systems. We don't have the data in those children under 18 or under 16, right? They have sometimes immature and strangely functioning immune systems till they get a little older. So that's a whole nother study that has to be done. And those studies are starting to happen. I've seen reports of kids who are like excited about being participants in clinical trials, because children really represent biologically, as that relates to the immune system, right? We all know, like, I don't get kids, right? Right? They are just very different. They're not just small adults. They immunologically are different. And so an entire study has to be built around studying the safety and efficacy in children.
So this, I'm not sure if this is a question, but it says the CDC is recommending use of the V-Safe app to track vaccine recipients, including the side effects. And then it gives the URL for that.
Yeah. So I'm not familiar with that, but I think one of the things that our public health enterprise, so remember I said one of the facets of our public health or our pandemic response is our public health enterprise. And so I think CDC is taking a proactive role here in providing a mechanism for people to report their experiences with their vaccine, right? So most of us will experience the first time around a sore arm. And then on the second dose, you may have more extreme reactogenic experiences. You might have fever, you might have muscle aches. So they want to give people a way to report that. I think that it has the possibility to impact the delivery or the deployment of the vaccine. It's also just collecting information about the kinds of responses people have physically to being vaccinated. And it's valuable information to have. We may, the short term, the phase 3 trials only spanned a couple of months. And so having longer longitudinal data on vaccine responses is incredibly valuable.
I don't know if your students ask this many questions, but
oh, they do.
they do. Can you explain why the standard method, such as attenuated live virus vaccines, does not work for coronaviruses?
That's not to say I'm sorry, I didn't mean to give the impression that it does not work. There are, in fact, vaccines in current clinical trials and actually have been approved in China that are live attenuated coronavirus. So they've been approved in other countries, namely China. But that is a strategy, I think, if you sort of evaluate the potential safety risks when you have all of these platforms, nucleic acids, protein, vectored vaccines, and live attenuated vaccines, in terms of safety, right? In developing a new vaccine, if you have your wish list, then live attenuated vaccines are probably the least attractive because of the risks associated with live attenuated vaccines, which is that they could possibly cause disease.
Okay, and I read this question wrong first. This says, what is your Zoom background? And I thought they meant, what experience do we have with it? But I think they mean, what is that background behind you?
I'm very
And
experienced in Zoom. I am standing in front of some art by David Goodsell, who is a talented molecular biologist and artist who does very detailed paintings of molecular locations. So I am inside a red blood cell, and what you're looking at are the hemoglobin molecules that carry the oxygen in your red blood cells. That's the painting. Yeah.
That's very nice. And so I was going to ask a similar question that we asked many of the faculty. And so could you tell us a little bit about teaching your classes, either in the fall or now, in person, virtual, and how's it going and yeah.
Well, so as a biology instructor, right, I virtually every class I teach has a laboratory, and learning biology in class and lecture is effective, but it's really enhanced by being able to be in the laboratory experiencing some of the scientific techniques that you're learning about in your class. So both in the fall and now in the spring, my colleagues and myself have worked very hard to be able to deliver laboratory experiences to students, which I think, even though we're restricted in many ways in our teaching because of COVID, working so hard to be able to do that, I think has helped students. It's been valuable for students. It certainly makes my life way more fun. I couldn't imagine like teaching labs from Zoom. It would be impossible. But we've been, I think, really successful with students under these COVID restrictions to be able to still bring them a really rich scientific education experience. So I've been really grateful for that. I I feel very, very fortunate that I get to meet with all of the students in my class at some point during the week. We're not at full occupancy of our rooms or our labs, but I get to meet all my students. And so that's actually, it's eased the burden of teaching in the pandemic quite a bit to be able to still be able to do that as hard as it is. It's good for me and it's good for our students.
And then this is going back to the other questions about your talk. What do you think the future is a year from now? Are we every year going to have a COVID vaccine where they've tailored it kind of like the flu vaccine? Or are we going to every year have to have two every 4 weeks apart or whatever?
Well, I think SARS-CoV-2 is with us for a long time. We will not be able to eradicate it in the next year or two. So we are looking at really needing to develop. We've all heard about herd immunity, right? And so we're really going to need to develop herd immunity. And but the virus is not going away. It might be reduced to seasonal like outbreaks like our flu season is. But I think it's unlikely that we're going to eradicate it in the near future. And so vaccination is going to be a continued feature. And I think you were asking questions earlier about like, well, how long does the immunity last and how effective is it, a year or 2 years away? And we haven't been in the pandemic we haven't been post-vaccine in the pandemic that long. So those questions remain to be answered. They're really important to be answered. And I think until we get those questions about what we would call the durability of the immunity that generated by the vaccines that are coming out, we'll have a better sense of what the end game will look like. But I think for the short term, COVID-2 is with us for several years to come, and it will probably be reduced to a more seasonal kind of outbreak if we can achieve enough herd immunity.
So this question asked twice, I don't know, but does the mRNA encode only part of the spike protein or the whole thing?
I'm sorry, can you, does the mRNA?
Does the mRNA vaccine or encode only part of the spike protein or the whole thing?
For the mRNA vaccines, it encodes the entire spike, and then the spike protein actually assembles in a multi-protein assembly in the cell membrane or in the membrane of the virus, that greasy coating. So that encodes the entire sequence of the protein, which then also assembles into these bigger structures that are presented to the immune system.
This sounds somewhat like Dr. Atlas or something, but many people think that getting the virus and surviving is just as good as getting the vaccine toward herd immunity. And why is this faulty logic?
Well, I think it comes down to this idea of surviving, right? We all think, I'll survive, right? We're all terrible at assessing risk. And so, and we know, because we know that many people experience mild COVID, and, you know, maybe even if they get sick and have to be out of work, they recover and they have little consequence of that. But the truth is that there are cases where there are severe consequences with coronavirus. And I don't think that's a risk worth taking, right? My risk of getting of those severe consequences is really unpredictable at this time, as my risk is whether I'll have no consequences of a COVID infection. So my personal feeling is I don't want to run that. I'm not willing to accept that risk of the really negative consequences of coronavirus infection. When I don't have to, right? There's no reason I have to, because a vaccine will provide me with the immunity that infection would. And I have not zero, but virtually, I have very little risk for severe consequences.
It reminds me of when I was small. If somebody in the neighborhood got measles, all the children would be sent over there so they could get the measles or chickenpox, and that's how all children seemed to get them. And but it wasn't that 1 out of 5 children was dying from the measles at that time.
Right. So, and that's true. It's not, but the 1 out of 20 or 1 out of 50 might end up sterile or have severe consequences of measles. And so why take that risk? Why put your community at risk for that and your own family at risk for that when you can achieve the same thing with a vaccine and not have the disease at all? So that's kind of how I would argue that.
I'm going to ask maybe one more question here. If you don't know if the vaccine prevents an asymptomatic individual from spreading the disease, could you please explain how it helps us achieve herd immunity?
Okay, well, let's see. This is kind of a complicated question. So herd immunity is something that we talk about. So we are the herd, right? The human population, the US population, right? That we are the herd. And it is important in limiting infection that a certain proportion of the herd possess immunity. Now, you can possess that immunity because you got an infection, or you can possess the immunity because you got a vaccine. And in my estimation, it's more desirable to possess the immunity because you got the vaccine. What then herd immunity affords us, right, is that it breaks the chain of transmission. So even if I am vaccinated and not susceptible to disease, but I can mildly transmit to people. But if I'm surrounded by vaccinated individuals who are not at risk for disease because we have achieved herd immunity, that's overall better for our population health. So I want to say that the idea that we might be transmitting virus when we are vaccinated is different from the idea of a sick person transmitting virus. There are a lot of variables that we don't yet understand. So it is formally possible for asymptomatic individuals to transmit virus, and it can make someone else sick. But exactly what that takes, and sort of what is going on with their immune response, these are all questions I don't have really good answers to. But so I would go back to this idea that essentially what herd immunity does is help break the chain of transmission for for disease, right? And so if in our community we're all protected, it's not a huge consequence to have transmission at whatever level it is and whatever that might result when most of us are immune. So I feel like that's not the best, most satisfying answer, but we understand so little about what asymptomatic transmission really looks like that it's hard for me to answer that question.
Here's another question. I think I said we were almost at the end, and maybe we are, but I know two people, now that's not me, that's who's asking the question, who had COVID twice and were much sicker the second time. Why is that?
Well, there could be a number of things. One simple explanation might be that, sometimes we have immune responses that are more or less severe, right? And so I wonder if in these individuals, the first time that they experienced COVID, their infection kind of ran the normal course of infection. And then they developed an immune response to it. And you would think, well, that should be protective. But it would appear to me, since they got it again, that their immune response actually wasn't appropriately directed. And so perhaps instead of being appropriately directed at controlling their infection, maybe it exacerbated the infection. That I'm really just speculating there. I think, I mean, I've heard these kinds of anecdotes where people get COVID a second time, and there's real questions about what is their immune status? Why wasn't that primary infection good enough to, or appropriate to educate their immune response? So I suspect there's other things going on there that I can't, it's hard for me to speculate about, but I think this is this is not unheard of and is something kind of maybe unusual and unique to COVID that really needs some research attention.
So now kind of a question for you. You've been a professional studying viruses for, let me say, quite a few years. I can't add that up. Is this the greatest thing that's ever came along for you? By that, I mean intellectually stimulating, watching the vaccines be developed and all that. Is this?
Yeah,
Is
I
this
mean, I think it's the first time in my scientific career where I've been really able to kind of, as a somewhat insider, look at what's happening to us and frankly be disappointed and dismayed, right? I go back to early in the outbreak where we had a lot of problems developing coronavirus diagnostics. And I, as a scientist, I understand how those diagnostics work. I'm not necessarily equipped to just go to my lab and, do coronavirus diagnostics, but I understand how they work in principle. And so, my scientific training, contributed to, for me, a lot of frustration and just, irritation about our scientific enterprise, why we couldn't get it together. Why do we have so much trouble with diagnostics? But it also, yeah, I mean, I said this before a couple times, like, it's a good year to be a virologist. I really feel like I can share my expertise with my friends and family and my community, and I'm grateful to be able to do that. And yeah, I mean, like, as I prepared for this talk, right, I'm not a vaccinologist. I'm really a molecular virologist. And so being able to dive into, sort of the manufacturing processes and sort of the industrial processes that really are involved in bringing vaccines to market is incredible. If I had it all to go over, if I could do it all over again, I would think, oh, I would love to do that, right? I've learned so much, just keeping up to speed on the things. And then the flip side of that is that I can't keep up with everything. I feel like, every day there is some new report or something new that is like has flown under the radar. I can't see it. So that it's also, while it's invigorating, it's also frustrating.
Well, with that, I think we're going to wind it up, and everybody out in the audience is cheering and clapping, but you can't see
Thank
that.
you.
But we're getting a lot of comments on what a great talk this was, Caroline. And one of the comments is one hell of a talk. Well, that's using the word as a superlative, but it's just a great talk. So much information and so well presented, and we thank you so much. we're so impressed with the faculty at FLC. So thank
Well,
you.
thank you very much. I was delighted to do it. It's just really a pleasure to be able to do this for our community. So thank you for having me.
And, you know, maybe we'll look forward to having you back, not a different COVID, but maybe a summation speech where it's completely gone. Herd immunity is here. Does anybody remember COVID? Oh,
Right.
my.
That I look forward to that day. I will be
You look
happily give that
forward to
talk.
giving that talk. Well, I'm going to end the broadcast. All you folks out there, you know, this has been recorded. It'll show up on our website tomorrow. So either you can come back and learn more from this talk, or tell your neighbors and friends to come and watch it. And thank you so much for the talk, Caroline. It's
You're very welcome.
lovely, lovely,
Thanks.
lovely.